Perfect. Yeah, good afternoon, everybody. My name is Thomas Hagan, and today I'm here to talk to you about the work I've been doing, as Nate said, on founder effects and how they reduce fitness at the range edge of an expanding invasive insect population. I'd also like to begin today by acknowledging the traditional custodians of the land in which this research was performed, the Jabogai, Juru, Idingi, Mbabarum, and Kuku Yalanchi people, and also like to acknowledge the traditional custodians of the land in which I talk from today, the Gadigal people of the Eora Nation. I'd like to pay respects to their elders past, present, and emerging, and extend that respect to any Aboriginal or Torres Strait Islander peoples present today. So I guess I should really start this talk with what founder effects are. Founder effects are the often large loss of genetic diversity that occurs when a small number of individuals found a population. So shown on the figure here on the left, each colored circle represents a different allele, and a native population is generally speaking genetically diverse and high in allele richness. However, during a founding process, where a small number of individuals with a limited gene pool found a new population, the resultant population, once it bounces back, will have a limited gene pool and be low in genetic diversity and allele richness. This can have fitness effects, but they are often difficult to ascertain. And maybe what is even potentially more interesting is that this can also occur during range expansion or range dispersal. As the population spreads outwards, the populations at the expanding edge tend to be small and subject to significant genetic drift and founder effects. The effect is compounded over generations as long as the expanding front continues expanding. This now brings me to my study species, Apis cerana. Apis cerana, or the Asian honeybees. Asian honeybee is a species of cavity nesting honeybee that is native to a large portion of Asia, but has become invasive in Australasia in recent years. The first major invasive population was in Papua New Guinea, where the species became invasive in the 80s. And from that population, a newly invasive population in Cairns was founded in 2007. While hard to ascertain information about the Papua New Guinean population, we know that the Cairns population is likely about 20,000 colonies strong at this point, and likely started from a single colony headed by a multiply mated queen. This means the population is quite low in genetic diversity, but why does that matter? Well, it's because of Hymenoptera's weird sex determination system. The basal form of sex determination in Hymenoptera is what is known as complementary sex determination. Under this system, sex is determined by zygosity at one or a few loci. And in the case of honeybees, this is one locus that is fairly well characterized, called the complementary sex determiner, or shortened to CSD. If an individual is born from an unfertilized haploid egg, they are hemizygous at CSD and develop as a regular haploid male. However, if they're born from a fertilized diploid egg, they could be heterozygous or homozygous at the locus. If heterozygous, they develop as a regular female, but if they are born homozygous at the locus, they develop into a diploid male, which in honeybees are inviable and lead to fitness consequences for the colony as a whole. Generally speaking, native populations of Hymenoptera have a high number of sex alleles that are more or less balanced in equal frequency. Therefore, Apis cerana and honeybees in general are excellent candidates to study founder effects, as the results of low genetic diversity are clearly linked to fitness at this locus and easy to quantify through genetic means. So given that, we had a few aims in studying this population in Cairns. First, we wanted to see if we could detect ongoing founder effect at range edges using neutral variation. And then, of course, we wanted to see if we could do the same thing using the sex locus, CSD. Then we wanted to know if diploid male production was higher at range edges and whether, and this is what we would expect from significant founder effects. And lastly, if we did detect higher diploid male production at range edges, if that was then slowing the dispersal of the population down. So to answer these aims, we collected samples with using two main methods. The first I'll talk here now is the collection of drones, which are the males. We collect them at drone congregation areas. This is done fairly simply. They meet in specific areas at specific times of the day in attempts to mate with queens, which they do about 6 to 8 meters in the air. We take advantage of this behavior by attaching a balloon to a fishing line, affixing queen lures to this line and luring the drones in, and they get caught on this trap as it's coated all in a sticky glue. As you can see from the video, depending on the weather conditions, you're sometimes able to collect a few hundred drones in as little as 2 hours. So after collecting the drones, we subdivided the population into geographical areas for the purposes of our analyses. This is a map of Cairns where the accepted range of the population is highlighted in yellow and each subregion is in a different color. Subdivisions were made mostly on geographic grounds, with one exception being the Southern Edge subdivision. We placed this as its own subdivision, as here we found drones outside the reported range of the species. After finding these drones, we then genotyped them for both SNPs and at their sex locus. We then performed a PCA using SNPs in all regions except West. There are three major points from this PCA. First, most drones cluster very tightly in the middle of the graph, regardless of region. This tells us, for the most part, they're fairly similar. Secondly, the components are quite small, and there's not that many SNPs that compose this PCA either. So there's not that much variation in the population. But what is interesting is that you'll notice that some individuals from the northern region and from the Southern Edge region do differ from the norm. Most, however, even in those regions, still cluster in that center bit. So we're able to tell differences in SNP diversity at these sites, but only for some drones, and overall, it's really quite small differences. So when it comes to neutral variation, there's very weak evidence of differentiation at range edges, but overall, we can't really tell too much. But what does the sex locus tell us? Well, we looked to see if subregions had significantly different diversity than the total population at the sex locus, and to do so, we used a modified bootstrapping method. So for this bootstrapping method, we first calculated haplotype diversity, which is the haploid analog of heterozygosity, and we calculated that for the sex locus of each subpopulation. The pie charts featured here represent the frequency of sex alleles in each subpopulation. Second, we found the frequency of each sex allele for the entire population, and from that distribution, we randomly sampled the same number of alleles in each subpopulation from the total population distribution, each time calculating haplotype diversity. We then repeated this sampling process 1 million times to get a distribution of haplotype diversities, much in the same way that you would maybe do with the Fisher's exact test. We then compared the observed haplotype diversity, which is the dashed line on these figures, to see whether they lay within the most common 95% of values in the distribution. And if not, we considered the subpopulation to be significantly different than the total population. From this analysis, we found that the central, western, and southern subregions did not differ significantly from the total population. However, once again, we found that the northern and southern populations did, and quite strongly. We also found much smaller scale differences in allele frequencies, but I don't have the time to be covering them today, unfortunately. So in contrast to neutral variation, we see that the sex locus does show significant founder effects. But are these founder effects linked to diploid male production and therefore linked to fitness consequences? Well, to find this out, we actually use colonies rather than drones. Through examining a colony's workers, we're able to calculate the amount of diploid males produced by a colony as a proportion of brood. And the way to do this is fairly simple. Essentially, if a queen mates with a male that shares one of her sex alleles, she produces a regular worker that shares her genotype and a corresponding diploid male in a 1-to-1 ratio. Therefore, by counting the number of workers that share a genotype with the queen, you're able to find the proportion of brood that would have been diploid male if they weren't endiable and dead. So after finding this proportion, we plotted this against the distance the colony was found from Cairns. This distance is proportional to the range edge at time of catchment, which allows us to look at colonies that have traveled a bit further on and past that point and push the range edge out a little bit. We found this relationship to be significant. You can see that the results are pretty variable, and we sometimes find colonies that have a high proportion of diploid males in the center of the range. However, the colonies found close to the range edge or further out than what we expect always have a high proportion of diploid male offspring. This is exactly what we would predict from serial founder effects. And furthermore, we decided to follow this up with simulations to see if this was true. And the model we used consisted of a 3-step process: a reproductive stage, a movement stage, and a selection stage. We let this simulation repeat itself for 20 generations and analyze the diploid male production of each colony and its relationship to distance. And as you can see, the exact same pattern is reflected in these results as well. This was modeled with several varying parameters, but most show the same result. Interestingly though, if you limit the dispersal rate of the population, that is, it moves out slower, the proportion of diploid male brood, while still following the same trend, generally has a smaller effect. So yes, diploid male production is higher at range edges and higher than the average amount of diploid male production in the range center. This therefore has fitness consequences, but does this result in a slower dispersal rate? And to do this, we once again look back towards our simulation. So our model from earlier doubles as a way to look at how populations can expand. And on screen here is a histogram of the positions of all the colonies in our simulations for our simulations that had limited dispersal rates. And this is a histogram of colonies for our simulations without that limited dispersal. So it's allowed to disperse at the maximum possible rate. And you can see that the edges get a bit further out in these simulations than with the limited dispersal rate. We then repeated this and ran a few simulations with the high dispersal rate again, but this time unlinked the fitness of a colony from the sex locus, such that the fitness is always equal to 1. Essentially allows us to look how far colonies get when they're not limited by a gene under balancing selection. And they get even further. They get a pretty far distance out. And if you look at the maximum distance that each simulation gets, we see a significant relationship. It's significantly higher than either of the two previous conditions when they are not limited by this gene under balancing selection. We also looked at real world data that shows basically a similar effect, but unfortunately I don't have time to go through that today. So now we've answered our final aim, and yes, it's likely that fitness consequences of low diversity and founder effects are resulting in a slow dispersal for this population. So to sum up, the population is dispersing slower than we would expect. This is likely due to increasing rates of inbreeding depression at the expanding range edge, a result of low population density and of founder effects. And lastly, this result probably wouldn't have been detected without the use of an adequate sample locus. Neutral variation alone would not have detected this effect. And lastly, I'd just like to acknowledge a few people. Obviously, my supervisors, Roz and Ben, particularly Roz, the many volunteers I had that came up to Cairns with me, Queensland Biosecurity, as they were always very nice in helping us collect samples, and obviously the entire B-Lab, who has been tireless in their efforts to help me with this research.
Nice. Excellent, Thomas. Yes, for those who don't know, Ben and Madalena have just retired, so it's an end of an era for honeybee research in Australia. But Roz and Emily and a few other folks will carry the mantle. Now, any questions for Thomas? Got a couple of minutes. Just sing out if you have one. I wanted to ask quickly, Thomas, any implications for how to control invasive species? Because obviously they do, so they go out and at the edges they've got to wait for some more to come. You know, so eventually they keep moving, but use this knowledge.
Yeah, unfortunately, we've actually done a bit more research that I didn't present here about how, well, specifically for loci that are under balancing selection, that tend to be under high mutation rates, which not all of them are, but some of them are. It seems like given time, they can actually restore that diversity, and in a relatively short time frame too, sometimes as little as 50 to 100 years. So while it does seem that loci with balancing selection are maybe able to slow down population expansion, it's not something that you could feasibly rely on to prevent invasive populations.
Good. Thanks for a very informative talk. Lots of nice modelling in there. Okay. Next up we have